Okay, great. Hello, everyone. Welcome to today's Journal Club. This Journal Club actually marks our second year of offering this opportunity to healthcare providers. We'll be briefly discussing a clinical trial funded by the FDA that evaluated CBD and liver enzymes. The next slide. Yep. So today, leading our discussion is Dr. Phil Molloy, a rheumatologist, and he continues to be very active for caring for patients despite retiring. Dr. Jahan Marcu is a cannabinoid researcher and the owner of Delaware Analytical Research Lab. And Dr. Len Kamen, the clinical director at Moss Rehab Outpatient Center at Jefferson University. He's also the president of the Pain Society in Pennsylvania and the head of the Rothman Opioid Foundation. Next slide. Okay, so for our first time attendees, I'm the CEO of Physicians Research Center Plus, and this is the Journal Club is brought to you by them. There's three areas of expertise. Salus is our phase 2 through 4 clinical study pillar. Buds Info is our analytics pillar, and Cerium is our education pillar. Cerium is designed for the busy healthcare provider who wants to understand cannabis and its derivatives through the lens of science. Next slide. Right now, if you are new, and we would like to thank you for taking the time to join us today. And if you could scan the QR code and participate in our ongoing survey, which is actually designed to understand you who participate. And then we can continually improve our offering. So, so if you have a minute, you can scan that. And our website is listed here on the bottom if you want to learn more at prctrialsplus.com and access our free educational blogs on the site. And I think that there are links, Jahan, now, right, to the YouTube?
Correct.
Yep. All right, next slide. So now I'm going to hand it over to Jahan to kick us off today.
All right. Hello, everyone, and welcome to this month's Cerium Journal Club. As you can see, this is our mission and vision page, and we support the education of healthcare professionals with discussions of published research, and we do our best to approach each paper with consistency, fairness, and transparency, even if we don't agree with it. And in spirit of that, we like to emphasize that the purpose of this journal club is to educate, raise awareness, engage in professional exchange. We're not recommending policy, regulatory decisions, or healthcare decisions, and really have no skin in the game or agenda other than education. And so sometimes we talk about articles that are controversial just to have those discussions, because I personally believe that through kind of this discomfort and exploring maybe data or things that disagree with what you see is a great way to learn, either to reinforce your hypotheses or to adapt them. Please share this link and this announcement, or however you got here, with colleagues. It's the same link every month. It's the first Tuesday of the month. So today's featured article is entitled Cannabidiol and Liver Enzyme Level Elevations in Healthy Adults, a Randomized Clinical Trial, published in the Journal of the American Medical Association. It has been available online since July 2025. So this study caught our attention because it seems to have some real world applications. It is a prospective RCT, not too many of those floating around with controlled cannabinoid products, and it addresses a clinically important issue. I'll go through the timeline, then I think I'll hand off some of the public health stuff to some other folks. uh, so how did we get here? Well, cannabis is prehistoric. There's lots of use. Ancient societies didn't really keep track of adverse events too much, so we have a bit of gap in information then. Uh, we 1960s really saw the dawn of modern scientific understanding. Indeed, gas chromatographs and the ability to even reliably measure potency didn't come around until then. 1970s, we had DEA drug scheduling and all the fun circus that is. And then in 2018, the Farm Act removed hemp and CBD from the this scheduling categories. And currently, there is widespread availability of CBD products online, retail, derivatives of these products, formulations of these products. Arguably, there's more variations of CBD products than of the entire library of products that the FDA regulates in the United States. Um, and we've also seen the FDA approve a prescription form of CBD. So, why do we care about this article? Because there exist gaps in knowledge about the benefits and safety of cannabis, the species, products, and all the things that can be made from it. Some products we understand better than others. And I think there's a lot of questions about these widely available CBD products. And there is a big public perception that there are many benefits of these products, and for having minimal, if no AEs. It's all safe in every form that you use it in, with little to no risk. But again, when we talk about hemp, we're talking about one of 100,000 or million products of formulations and variations thereof. So,
Nice.
so Teresa, I know that you have a background in public health epidemiology. It would be great if you could tell us a little bit about the slide and what are we looking at here? This background on liver function tests and CBD.
Sure. Thanks, Jahan. So the most widely used CBD product that was indicated for um, epilepsy in kids is Epidiolex, and it was approved in 2018, and 900 patients were in the trials. Now, when you think about this, 900 patients is not very many patients. Over the years, we've done clinical trials with you know, many, like 90,000 patients where you can get a much better risk estimate on your adverse event profile than on 900 patients, if you think about it. The CBD that was tested, it's in the package insert, range from 5 to 25 milligrams per kilogram per day. So it's weight-based. They looked at transaminases that were greater than 3 times the upper limit of normal, and they observed 12 to 13% versus 15% in placebo. Now, the levels of upper limit of normal, most people, everyone probably on this call has had their liver function tests, old as SGOT and SGLT, now AST and ALT in your chemistry profile. And the upper limit of normal is generally around, 40, 30, depending on the test. And there's regular monitoring of LFT. So when you go for your metabolic panel and your glucose and your all those other tests, these two are also included. And in the package insert, patients, may be used in patients with pre-existing liver disease with dose adjustments. This is the Epidiolex package insert, and it's possible to turn urine cannabis test positive with Epidiolex. The next slide.
Can I interject something?
Sure,
That's
sure.
it's an unexpected finding that cannabis pure CBD products will turn your urine test positive because the all the immunoassays routinely used now to check for cannabis are THC is the main antigen. And that's a different topic and it's a long one. But the assumption initially was if you're taking pure CBD, your urine cannabis test won't turn positive. But in fact, it does many times, surprisingly.
Yeah, thanks, Phil. And now the
Yeah.
perfect timing for you to talk about this. You like
I took
this is your slide, your picture.
yeah, I took I took the picture, the illustration on the left, from a liquor store that I go to buy wine at, and you can't go to a liquor store without seeing CBD infused products right now. So on the right-hand side, the white background part of the slide, is epidemiology. Cannabis use is really common in the United States, 20% in a given year. 60% use CBD products, have used it, 29% regularly, and 10 or 11% have used it recently. And bottom bullet, everyone knows that already. CBD products are available online, and you can take them topically, orally, in oils, capsules, gummies, and now as infused drinks is an alternative to alcohol. So the next slide. So I will start just by painting a picture of how this study was done. They studied 201 healthy participants over age 18 and under 55. They didn't want to get into old people like me who are taking maybe other medicines that might affect your liver test. So they wanted, say, reasonably healthy people. It was a double-blind randomized control study. Double-blind means that the evaluators and the patients didn't know what they were taking, either a placebo or CBD. It was performed in a Wisconsin inpatient clinical pharmacology unit on day one. And on the first day and last day of the study, you were hospitalized for a bunch of metrics and measurements. And most of the study was outpatient. The goal of this study is, does CBD cause abnormal LFTs, liver function testing? And Teresa already mentioned what they were. There are two common LFTs. The abbreviations are AST and ALT. And the primary endpoint of this study was either one of them being abnormal, not both necessarily. The dose they chose was 5 milligrams per kilogram, body weight adjusted. And they were given it as a split dose, half in the morning, half of the evening, or a matched placebo in an oral solution. And the notes, I put C notes. It was just to remind me that this dose, 5 milligrams per kilogram of CBD, if you scan the literature, above that dose is commonly known, already reported in the literature, to be associated with abnormal liver function tests, and below 5 milligrams per kilogram is rarely. So this is on the cusp. This dose was chosen because it seemed to be a cutoff of exposure to CBD that was associated by other researchers historically with abnormal liver function tests. And the primary endpoint is either one of these two being 3 times ULN. Get used to that abbreviation. It's spelled out for you at the bottom, the upper limit of normal. So if the upper limit of AST is 40, that you're abnormal, you're in this database of abnormal if you're 3 times 120. So let's look at the next slide. That's how it was done. The first night and the last night, overnight stays in the unit. When you went home, the morning of the second day, you were given a 28-day supply. There were online text reminders and clinic visits, outpatient, not overnight, at the dates you can see, 7, 14, and 21. The last day was 28. That's when you spent another night in the hospital. You checked out on day 29, and you came back at washed out 7 days of exposure to nothing. So that's how the study was done. For those of you who are interested, you may not be impressed with the numbers, a couple hundred people, but it's pretty rigorous. I thought the study design, I'm proud of them, and kudos to them for pulling this off. Not easy to do this. Next one. There are typical inclusion and exclusion criteria. For example, you couldn't have liver disease, or you couldn't have hepatitis B or C, and that sort of thing. It's usually it's the typical ones that are in all these studies that assess CBD. There were endocrine assessments, which were not the primary endpoint. They had to do with sex hormones, thyroid, and so forth, and they were performed at the beginning and day one and the last day. And plasma levels of CBD, for what it's worth, were measured. We do know, by the way, that there's a vast interpatient, intersubject variability. We all take the same dose, ingest the same dose, and our blood levels are vastly different. And it was done by chromatography and mass spec. So sophisticated, reliable methodologies in the lab, unless Jahan wants to say that you trust these, sort of a standard. You're a lab guy, Jahan. You own a lab, after all.
Yeah, I think liquid chromatography is superior these days to a lot of other analyses. And I'll fight you to the death if you say otherwise. I just gas chromatography has its uses. Other methods have their uses and their applications, but I feel liquid chromatography has many benefits. So when I see LC, my
Yeah.
bias is to think that yeah, they use the best methodology
Yeah.
available at this time.
All right, let's look at the next slide. I think the next slide is, yeah.
Yeah. So
We
this
have to
is
spend a lot of time on this. I hope all the people who are tuned in today downloaded the article. So this is in there. The number of people who were screened, the number of people who were randomized. You can see here it is a 3-to-1 randomization. So 200 people randomized, and where they were, uh, where they ended up. Placebo, 50 got placebo, the rest got the active product. And the timeline on the bottom is the overnights, the 2 overnights, and the days they had to come back for blood tests, urine tests, clinical assessments, and so forth. And the evaluators and the patients were subjects were blinded. Go ahead, next one.
and the other thing I just point out that this is important, particularly B, the lower section, because it shows the timing, because the liver signals turn out to be fairly delayed, not sort of immediate effects that you see. So the
Okay, so
design, yeah.
before you throw the next slide, have the audience guess when. Let's suppose there were some patients destined to get abnormal liver function tests. Did it occur at 1 day, 1 week, 2 weeks, 3 weeks? What is a good time to draw that test? If you have a patient where liver issues are concerned and they start CBD, are you going to check their liver test? And if you do, you're going to do it on day 1, week 2, week 3. When are you going to do it? And the answer's on the subsequent slides.
All right, 1 second. I'm just going to create a simple test. We'll say one
Right,
day.
because is it cumulative, Jahan? That's the question. Do they think that it would be a cumulative effect, so over time, so that would take longer, or is it more immediate, more acute?
All
Yeah,
right,
I
so
mean,
one
there
day,
are,
1 week, 1 month are the choices.
yes,
All right,
Sure.
all right. Save, and we will. All right, and let me go, and we'll throw that out there. We'll launch our poll here. See if it shows up. There we go. Untitled poll number 24. When to check liver enzymes? 1 day, 1 week, 1 month, never.
it's important because there are other drugs. I'm a rheumatologist, as most people know, and there are like anti-inflammatories. The excess cardiovascular risk and GI risk, they begin immediately the next day when you start them. So,
Absolutely.
but
We
Yeah.
can
other
only
drugs,
we can only pick one answer though, right?
Yeah,
Yeah.
yeah, no,
yes,
but this is after baseline. So just to make that clear,
yeah.
baseline
Oh,
is
okay. All right,
assumed. So
after
yeah,
baseline.
yeah, baseline is assumed for
All right,
sure.
you can show the poll results and then we'll show the facts,
All
what
right.
this 200 patient
Yeah,
study
but there was
showed.
a question in the chat about, you would definitely do baseline and at like some point after
Right,
that. So I just wanted to be sure that everyone knows that we're
yeah,
assuming that baseline is was
right,
would be drawn for sure.
right.
Great point out there. Oops, did I close that?
Yeah,
Where's the poll? All right. Oops. Oh no, it's showing the answer. All right. We'll end the poll and hopefully it lets me share those
sure.
results. But it was, oh wait, where are the results? There they are. Uh, Share results. Boom. So 90% of people on the call said 1 month, or sorry, it's about 60% said 1 month, 30% said about 1 week. I really don't think there's any wrong answer here, but we'll see what the researchers did. And here we go. So
This is more, yeah,
yeah, this
more
is more. So this
demographics.
is table one, which answers the age-old question, who are these people? Exactly. And so it kind of basically confirms that we have a balanced group at baseline. Mean age was 36, 44% female, BMI in the over 8 range, very typical of the general adult population. So basically, no baseline liver risk imbalance to skew the results,
They don't
which may
show that though. But go ahead.
No, but it's in
They
the
don't show
paper.
that. They don't so we're just this is the baseline characteristics. We're just gonna go with it and keep moving. We're not gonna talk
Yeah.
about yeah, there we go.
So this is interesting, isn't it? This is the primary endpoint. And if you have the paper open, anyone, the primary endpoint is shown in the figures 2 and 3. The primary endpoint was who has liver function tests, AST or ALT, 3 times higher than the upper limit of normal. And it was 8% of cannabis users at this fixed dose. 3.3%, 5 subjects, were 5 times the upper limit of normal, and 2 subjects, 1.3%, were 10 times the upper limit of normal. It's generally felt that the more abnormal they are, the more your liver is being irritated. And the bottom bullet is the number of people who were predefined. There were definitions of when you had to stop the study, and 7 patients had to stop it because of laboratory abnormalities. So this is the primary endpoint of the study you're looking at right here. And by the way, because it's not shown on this slide, how many placebo patients? The answer is 0. No placebo patients had abnormal liver function tests in this study. Well, let's look at the next slide.
so
This
this is table
this is the
two.
same results essentially shown in a table.
Yeah, this is a sort of a regulatory table that you'd expect to see in a paper like this. This is one like FDA reviewers might care about a lot or
Yeah,
something like that.
yeah.
Uh, Happy to come back to this slide if people have questions. So this
This
is, yeah,
interesting. Go ahead, Jahan. This is interesting.
so this is the Kaplan-Meier curve for the primary endpoint, and this figure sort of answers the question, when does the risk appear? So I think this is a fairly intuitive visualization. We basically see that the curve for the placebo is flat. Nothing happens. The curve for CBD stays flat until about day 21, and then by day 35, it has seems to have risen significantly, sort of crossed a threshold, if you will. So this figure tries to kind of tell us two things, that this effect may be CBD specific and it's delayed. Therefore, on my understanding, could be easy to miss in a real world scenario if we're talking about something that takes weeks to appear. Um, I think that this could undermine some short-term reassurances like I've been taking this for a week and I'm fine,
Yeah.
because it takes longer for these effects to happen. And
The, ne
so,
the
yeah,
next slide shows individual patient data. So here are 8 primary data for 8 individual patients. So each of these blocks is one patient, and it supports the previous slide, what Jehan was pointing out, that nothing happens in the first 3 weeks. You can look at each individual patient and sees that it all happened.
that's an important point, Phil, and I just want to emphasize it that these figures show the biological heterogeneity. These are not averages. So you can see these sort of, some participants had modest elevations, others had dramatic spikes,
Yeah,
sometimes exceeding the upper
yeah.
limit of normal by quite a lot, 18 times.
I want to comment on eosinophils.
Oops.
Eosinophils are a subset of your white cells that are often associated with having an allergic reaction to something. So these patients that had abnormal AST and ALT most of them had abnormal or elevated eosinophils as well. And ALK-Phos and GGT are 2 other liver function tests. They were not the primary endpoints.
It's a great
Let's
point.
look at the
Yeah, I mean
Yeah,
it's the that immune mediated component. I think
right,
it strengthens the causality of this.
right,
And maybe it's not as simple as dose toxicity, right?
right,
And it does appear that it might be uh, reversible after stopping CBD.
right,
Um, these measurements appear to be going down. So
right.
good news.
Yeah, that's another important. Thank you, Jahan. The gray area is the exposure. So look what happens to everybody at the two top ones. For example, when you stop the drug, the abnormal blood test resolve. That is the point of what Jahan's just saying.
And we'll add that someone in the chat added that this is a summary of a clinical study. It's not the whole clinical study. Absolutely true. If you have access to the internet, you can download the entire article, and there are some additional tables on sort of supplementary tables. There's a lot of supplementary tables that you can access as well to dig
Yeah,
deep. So some,
yeah. This is
yeah.
from the literature. If you do an AI search on CBD and liver function test, it says around 300 milligrams is the cutoff, which I mentioned before. That seems to be around 5 milligrams per kilogram per day in most people. And what about the bottom three bullets? These are meant um, is on conclusions from the paper. It is nudging you to reflect. What do you think? Should we follow liver function tests in our patients on CBD? There is a perception among healthcare providers and patients that CBD is a safe supplement. If you assume you have a baseline, I think everybody knows now that we wouldn't do the follow-up at 1 week, we'd do it at a month or so. Have you recommended CBD to any of your patients? And if you do, or do you query your patients if they're on CBD? And I mean, it makes me want to monitor their liver tests. Just throw that out there as a clinician. Makes me want to look.
And I wonder how many people out there who use CBD products regularly are consistently consuming, more than a few milligrams a day. That are because I noticed that some of the beverages um, are sort of mild placebos. They have 5 milligrams
Yeah,
of CBD in them,
yeah,
or some
right.
really um, low dose, just to be a little tongue in cheek about it. And I wonder, of the people who might consume 300 milligrams in a day, how often do they typically do it if it's not on prescription?
Right.
Um,
So
If
Jahan
any,
and Phil, are these questions like to our audience? I mean, I think that maybe it would be
yeah,
interesting. I mean, because I know we each have our own opinions. I know I have my opinion, but before I share my opinion, I'd like to hear from, did you prepare another like little poll of,
no,
I
there's
guess
no
the
poll.
questions would be is, if they do recommend CBD, do they monitor the LFTs or
Yeah,
actually discuss with their patients? Um, because, what comes to my mind is so many people use acetaminophen, for example.
yeah,
And, when you're using acetaminophen, there's a, a, you're not supposed to go over, I think it's like 4 grams or something like that a day, because it's, metabolized through the liver. And if you're using CBD and acetaminophen, like what happens? And CBD is not, you know, when it's on the shelf, it's not by weight like children's Tylenol is, or
right.
any of other childhood medicine is by weight. And for adults, it's not that. And so what they're taking, the 90-pound year old, the 90
Yeah,
pounds, 70-year-old is taking the same amount of CBD per label as the 200-pound,
yeah.
60-year-old male. So I think that that's a concern that I would have, and I'd like to know about the practitioners on the line. Like, what do you do if you're willing to share your thoughts?
yes, please feel free to unmute yourself now if you'd like. We only have one rule, and that is thou shall not journal club and drive. So if you are able to um, unmute yourself and you'd like to comment on these questions or any of these
I
prompts?
I have a comment.
Thank you.
Um, First, I think the cost of CBD alone is prohibitive for somebody taking a 70 kilogram person taking 300 or 350 milligrams a day of pure CBD is it's going to be highly prohibitive on a regular basis. It may be an episodic basis. Most of the formulations, I think, are typically, you know, 25 to 50 milligrams at most, and that's a high dose. I think the Charlotte's Web company makes a more potent CBD product that's like I've had some patients purchase that. It might be 100 milligrams, but I do think that the practicality of this is that most people do not ingest 350, that's a 70 kilogram person, 350 milligrams a day of CBD would be exorbitantly expensive.
Great point. And that maybe price is a public health leverage, maybe.
Mm.
But I think absolutely right. Yes, Jane, please go ahead. Thank you for joining.
Yeah, hi, Jahan. It's been a few years, but I'm still with APA. So, and I'm with the American Herbal Products Association, so a trade association, and we do represent people that make CBD products, and very much echo the other comment about just the cost prohibitive nature of taking this dose on some sort of consistent basis. And we've been looking at this study too and trying to reconcile some of the conclusions. And I think a question we came up with is what is the relevance of this study, which used Epidiolex, to the CBD products that most people are accessing, which are much different. They're not CBD isolates necessarily. They're Jehan, you mentioned that there's a variety of formats and different types of extracts and whatnot. So um, that's another thing to think about is this is not necessarily reflective of as a test article of what the majority of people are actually ingesting. Um, and I have a background in public health as well, and I think they missed an opportunity to not do multiple doses because it would have been interesting to see if there was a threshold where they didn't see this effect with some of the liver enzymes. So I know that's a much more complicated study and whatnot, but it seems to me like it would have been helpful to have a couple of doses less than this. We had some dialogue with FDA about this study, and we were asking how they settled on this particular dose level. And they actually based it on some market research from the Brightfield group. It's one of the references in the study.
Hmm.
And so we actually talked also to the Brightfield group about it. And so they had done this market study asking CBD users how much they take on a daily basis. And this was at the upper end of the responses that they were getting. But there was also some question about whether users were misinterpreting the total amount of CBD on the label as the dose that they were taking, so that they might have responded to this market information in a way that overestimated the amount that they were actually taking as a daily dose. So, we had a lot of questions about it. Obviously, we're happy to see this type of research out there, but sometimes it raises more questions than it ultimately answers as well. So. But thanks. It
Thank
was just
you.
great to sit in on and
Yeah.
listen.
Thank you, Jane. Great comments. I particularly love what you said about the labeling. I definitely have even though I've looked at labels for years, sometimes I'm struggle with it when it says 1,000 milligrams, and I'm like, is that in the entire bottle? Is that
Yeah.
per capsule? And to your point about the dosing, I think that's incredibly valid. Maybe not, dosing of the prescription CBD. That's quite expensive to add to an arm, but would be maybe less cost prohibitive for a study is having a group of people that use a CBD product of their choice. And they've done these sort of ad libitum or bring your product for the clinical study projects in the past where
Right, right. Yep, there's been at least one of those.
and I think that might be it would be very interesting to say, okay, we'll have this group of people who are using commercial CBD products compared to an isolate and see if the results are similar. You know, you have all this very variability in the market. I think it's a good point. Do the other cannabinoids when they're with CBD play nice, or do they cause a similar effect? We don't know. Great
If you
comment.
do a study, if you do a study like that, Jahan, it would be interesting to test the products too. Is what is actually in there, what is on the label?
And that brings up, well, how do we define these products and what are the cutoffs for ratios? And if it has, 0.5% CBC, is that enough of a difference to call it a different product than one that doesn't have 0.5% CBC or another
Yeah.
cannabinoid? But I would love to participate that. Anyone on this meeting that is running a clinical study, feel free to reach out. happy happy to test some products for a clinical study. Um,
Can I make, this is Tim McCarthy. Can I make a
oh,
quick point?
please do. Welcome to the Journal Club.
Good to be here. Thank you. I have studied this paper rather closely, and I've talked to Jane a little bit on one of the conferences last week about CBD in general, and um, I've talked extensively about this, and even talked to Norm Birnbaum about it with the FDA. Um, I think I have multiple years as a pharmaceutical sales trainer in oncology and had to review papers ad infinitum on many different subjects and had to go diving deep on, is this a real paper or is this made up for a title more than an outcome-based review? And I believe it is that one, and I'll tell you why. For one, the Brightfield group, the dose that they picked is the upper 11th percentile of the study. Almost everybody else is below that, but that's the one they picked, and they called normal. I don't know of anybody who's taking CBD isolate that, and I noticed the weight, the average kilogram was about 80. That's a 400 milligram dose, maybe 200 BID, but that's 400 per day dose. I don't know of anybody taking that, especially if they're taking it with multiple compounds. Most of the people in the trial were taking somewhere between 30 and 50 to 70 in the upper edge of CBD per day. So I think that's one thing. And then one thing that Graham and I pointed out to Graham, he's president of APA, is that he found me the actual protocol that this trial came from. And so besides the Brightfield group, which didn't do that on their own and just presented, it was actually sponsored by the FDA.
Hmm.
The other part is on the protocol, and you have to dive deep, and it's only mentioned on one page of 178 pages. And if you have pain, this is an 8-month trial. It ended up taking 8 months to accrue everybody and get it. So you're going to be going through at least 3 different seasons in 24. And if you're going to have a pain of any type, you're only allowed one pain medication, and it happens to be 1,000 milligrams of acetaminophen. That's all you're allowed by this protocol. You don't think that over 3 seasons out of 150 people that they wouldn't also have taken some Tylenol because it's so safe um, in this amount of trial? So I think the percentages they came up with, the 5 milligrams per kilogram, there are trials showing that elevates ALT already. That didn't go past their knowledge. They know exactly what that is. And to assure that they had an outcome to say this is a normal dose of CBD in the marketplace that people take, and this dosing raises ALT, but they don't put anywhere in this summary of the study, this is not the actual study, this is the summary. They don't mention anywhere in there that the only protocol that they allowed for pain was Tylenol, which we all know is liver toxic and the number one reason for acute liver failure in this country, needing for a liver transplant. So I think it's made for um a headline because no one's going to read 178 pages of the protocol. But that's what I did for a living for about 7 years. And I go through that level and find things. And that's how I was able to knock out competitors' comments or work it against them. Anyway, that's my 2 cents. I don't think a lot about this paper as far as something to tell us that we have to worry about because I don't think the range is typical for what a patient would have. All of this was subclinical. Nobody complained. The very few people complained, and they got off of it. But at that dose, if I was 80 kilograms, I'd be taking 400 milligrams a day. I don't know of anybody, and I've been selling cannabinoids for, since 2020. I don't know anybody taking that kind of dose unless you're a child with epilepsy. My 2 cents worth. I'll shut up now. Thank you.
Thank you, Tim.
Thank you very much.
That was great. And I think you're right on target with dissecting the study and clarifying the results. So that's thank you so much. Um, I definitely hope you show up to more journal clubs and because we try to go through a lot of picking, for lack of a better term, some uncomfortable or discordant research and walk through it and discuss it. And we don't have all the answers ourselves. And sometimes we don't want to read the 200 pages of background either. And so it's great to put these things out there to the community. I think that's fascinating about the Tylenol aspect. And I think it's always good to be reminded that sometimes people are really driven to publish a result and a striking result. And the study is sometimes designed to say, see, this can happen under these conditions. This can happen some of the time, but it's not necessarily the rule that will happen to everybody who's exposed to a CBD product.
Right,
But
and this is Teresa,
But
and
it's
I really
presented that way.
appreciate I appreciate your comments, Tim, because when I had made the comments about table one, I know that it was limited, but I couldn't find, and it's about other medications like over-the-counter medications like acetaminophen, because to your point, if you are in a long-term study like that, they have to allow something. And, to go through the patient level data to see, are those the ones that had the increase? I mean, those are the kind of questions that I have. I wonder like who the patients are. Are they different in some way? The ones that had the liver enzymes increases versus those that did not. But it's awesome that you spent the time in pulling the protocol and looking at all the supplemental information. Thanks for that. And same for Laura and Jane, all your comments. Very helpful.
And while you were talking, thank you. I want to add my thanks. I went online and shopped for CBD. Um, that dose is going to cost somebody $1,000 a month on the internet, depending on where you get it and all that stuff. But if you look at 5 milligrams per kilogram, or the upper limit of 300 or 400 milligrams total, that's the ballpark that patients are paying out of pocket.
Yeah, so the question that I have is that why, like, and Tim, maybe you could provide some insight since you spoke to Norm. Um, Why would the FDA fund a study with CBD at such a high dose? Like, why wouldn't we want to understand something that's a little bit more practical for the common consumer because they are consuming this product? I mean, I've worked in pharma for greater than 30 years, mostly in like not in oncology, but cardiovascular, rheumatology, all the immune response diseases. So I'm curious what you think about that, just your opinion.
Oh my, this is I should wear my tin hat to give you this answer. If you look at many of the studies that have been published about cannabinoids in general, but it's either THC or CBD in JAMA, you will notice almost every single one of them is negative in some way. I think JAMA would print a letter from the FDA or a study, because this study is done by the FDA for a ham sandwich and call it a monumental study should the FDA ask them to. So I don't think it's an unbiased scenario. Again, I'm wearing my tin hat right now. But I think it's a situation where if THC gets put down to Schedule 3 and goes from no medical value whatsoever to some medical value, the FDA would like to try to take over most all cannabinoids. And papers like this, where people are going to read the headline that a normal dose of normal dose of CBD is still causing liver problems. See, it's too dangerous. We have to be able to monitor this ourselves. It is not going to work right. We have to be the ones to monitor this. And I think Norm and his capacity, my conversation with him, Graham was right there to my left, Norm was to my right as we spoke to him. And he kept trying to say that we're open for multiple cannabinoids in a product. When I asked him, well, how about studies? Why did you pick just an isolate? And he says, well, that's the only way to make sure it's just that one product. And I said, well, no one really takes it that way. And if they take isolates, they're taking 10, 20, 30 milligrams at a time. They're not taking 200, 400 milligrams in a day. And he goes, well, that's the only way to isolate if there's a problem. And I said, well, you presented this um, as a study. Actually, I acted like I didn't know the study when I was talking to him, so I had fun doing that. I said, well, is this the normal studies? Oh, yeah, normal doses. Oh, absolutely, it's normal doses. We have a survey that told us that. That's why we picked it. I'm like, oh, really? So that was from the middle of the pack? Is that from the most typical, or is that what is the upper range? And he goes, well, we determined that that upper range should be considered normal. Oh, you picked that. Really? So I'm like, okay, well, what about if we have speaking of CBC, what if we have a CBD and a CBC combined? Well, that would be trouble trying to find out which the toxicities are possibly coming from. Oh, so then you only want isolates to study. Is that accurate? No real world studies like the executive order has asked for and alluded to in the Oval Office. And he was like, that's when he got uncomfortable and had to go.
Yeah, because they're trying to treat it like you would, as you know, a clinical trial where you do the components and the combination. So for that study, you would do CBD, CBC separately, and then the combination. And you know, it's amazing to me how the FDA for this product is it seems like I guess I just don't understand, right? So if there was any other product on the market developed by pharma, it would kind of like be slightly different because you would do all of those traditional trials. Not, like dose finding, you would do all of it in a sequential way, as you would know. This would be the like.
So we have to come down to what is going to be allowed. Is it going to be a dietary supplement type, or is it going to be a pharmaceutical type? And I don't just preach the choir here. I know I'm preaching to the preacher. I apologize for this, but um, it's no one's going to be putting this out as an isolate. No one thinks it works that well as an isolate. I certainly don't. I love combining it with other cannabinoids and other botanicals because I think it does play well with others. I think they're complementary. I think a little bit of CBC goes a very long way to being a multiplier for CBD. I think CBG can do the same things in a different way, and I love the forms of the acids that are in there. They work even better. There's a few people out there making products that have the A's in a salt form, and it's much more stable. So I think those combinations are actually, if I can't remember which one of you suggested it, but if we could actually get people to pull off the shelf what they're taking and bringing it in, and let's take a look at that. But the FDA is never going to do that. Norm is never going to allow that. Norm would like to have his own little fiefdom and control cannabinoids till he's tired of doing it.
Now,
Well,
yeah.
it's going to be interesting to happen with this pilot program through CMS, where they're looking at doing Medicare reimbursement for CBD products. And so it's really going to force the issue with FDA of what they're going to accept.
Well, hopefully it can get looked at under DSHEA. I know you and Appa are spearheading a lot of that, and I appreciate that work, by the way. But that's pretty much what it's going to have to take is either Congress, declare it that way in some form, so it's not
Yeah.
just going to be pharmaceutical. I mean, we have um, uh, 800 milligrams of I'm just forgetting my pain. I've got acetaminophen on my head so much. Ibuprofen. Thank you. You have a pharmaceutical dose, 800 milligrams of ibuprofen that's still an RX only. But meanwhile, we can go buy 200 milligrams with a bottle of 500 if we wanted to
Right,
from Costco.
right.
So,
And take the 800 milligrams. Yep,
and
exactly.
take the 800 milligrams anyway. So there's the over-the-counter dose and the over-counter type product or a pharmaceutical product. So where are they going to actually try to allow this to sit? And that's what I would be interested in, um, is having the discussion. Appa's much better at that. They have all the connections for that. If you ever want my support or my anything that I can do, I'll be glad to help. Oh, also, besides my pharma background, I don't do that anymore, of course. I am with the National Industrial Hemp Council. So we're the sole cooperator for hemp with the USDA. I have regular conversations with them. I know Appa does. I'm just saying what I do.
Well,
Um,
thank you.
Yeah, it would be
Thank you.
great to follow up. I
Yeah,
appreciate that.
yeah, I agree. I agree. And the attendees for this particular one, maybe it was the topic, um, because it seems to be of a lot of interest from a lot of groups, right? And this was extremely helpful. I know that we're at the top of the hour. Everyone has to go back to seeing patients or their next meeting. Um, who wants to wrap this up?
Uh, well, I will just summarize in saying I hope the FDA definition of cannabinoids goes beyond anything given to a human that results in a publication as a drug, and that they have a much broader or maybe real world interpretation of those products. I think the most important thing is that we're going to have another journal club. Tuesday, we're going to look at the ACOG publication about cannabis use during pregnancy and lactation. It's a topic that we've covered similarly in the past. Again, this is one of those headline articles where folks either talk about it incessantly or refuse to talk about it at all. And we want to break it down and walk through it with all of you. In April, we'll be looking at a review on ADHD and cannabinoids. And in May, we're thinking about an article about high potency THC products. Always fun to discuss and break down those studies. We're also open to suggestions for topics. So feel free to reach out through us through the website, share your email here, or just shout it into your microphone at any time. Uh, but we want to thank you so much for your time and attention in these long form discussions about published research. Great discussion today. I always love it when we don't even get through the slides because there's such great conversation going on.
sorry.
It's really our goal. So thank you.
Any more comments from Dr. Kamen or Phil, Dr. Molloy?
No, this study obviously has strengths and weaknesses that we're going to have to ponder. That's all as we move forward.
All
Long,
right.
I was in Manhattan last weekend, and you get out of the train station, and it just reeks of. And I was thinking, with this study, all those liver surgeons are going to do fantastic,
It certainly has improved the smell in the subway, I want to say. All right. Well, I want to be respectful of everyone's time, so I'm going to start to adjourn the meeting. Thank you so much again.
All right.
Thank
Thank
you.
you.
Appreciate
Thanks.
you having me on,
Bye.
having with you.
Thanks.
I hadn't had time to look at the comments. Sorry if I'm still on you.
Oh,
That's
no,
okay.
no worries. I'm going to shut it off in just a second, but take a moment. Um, uh, Tim, what's the actually? I'm glad you hung out for a minute. What's the best way to reach out for you? I've done with my PhD background in cannabis, I've done some like expert witnessing and regulatory stuff with like different groups, and would just love to chat some more.
Yeah, no, I'd love the correspondence. Whatever you need. So, I have no medical background. Everything is self-taught. Um, but I have ADD in the right kind of way in that if you've ever heard of hyperfocus.
Yeah, yeah.
So if I get into these studies in a particular interest, I can pound through some papers. And I can't always pick every last little thing, but from a marketing and sales standpoint, I had to notice what was the most important factors to either worry about, to either work a story against or use it for our advantage. So recognizing patterns is actually what I have gotten good at. And you can read a white paper dressed up as an actual study. That's what I think this one is. They knew the outcome they would get. They picked Tylenol to put in there and purposefully didn't let everybody know that it was in there because they know that would torpedo the study. Um, but that's the protocol allowed. That's the only one in it. I think it's page 28 of the protocol. Most of the last half is, references and studies, but it's just on one little small piece that, and not just Tylenol,
Yeah, is that in
1,000 milligrams,
clinicaltrials.gov? Was
the
that
upper dose.
yeah, was that in clinicaltrials.gov? Is that how you downloaded it?
No, not Norm. Graham got it for me.
Okay.
Graham found it and sent it to me because I'd been talking to because we had met at that in DC at that conference that they had on THC. Um, but I started talking to him about this trial because I knew it was absolutely ridiculous, and I know that the FDA is trying to work this to where they can put a bad light on CBD. And I just think that's bogus, and it's a control factor. I watched Amgen do it time and time again against other oncology products. So I've gotten used to looking and seeing this glaring at me. Um, and that's what this was. I mean, who picks the upper 11 percentile of a survey?
Someone who wants a publication.
And then who doesn't tell everybody that there's Tylenol allowed in the trial, a liver toxic drug?
Yeah,
I
I
mean, it
think
wipes
that
out glutathione. I mean, for days, a single dose. Suddenly, you're not going to have a lot of toxins being eliminated from your body. Acetaminophen is a terrible med. The only thing it's safe against was when it first came out, and I'm of this age, I remember all this. It was just juxtaposed against aspirin and the stomach problems. And they have sold that as their safest thing, but you know, really, it is if you know what glutathione does for you, and that's why it's liver toxic, it's because it totally wipes out glutathione. And the longer you take it, the longer it suppresses the glutathione in your body, and you need that antioxidant to remove other toxins from your body. So the next time you have a cold and you think you have a fever and then you're going to take this safe thing called acetaminophen, and you wonder why you still feel terrible, is because while it may eliminate the pain, it's making all the toxins build up in your system.
hmm, interesting.
And
Wow. So we could go on for another hour probably,
But
Tim, to chat.
sorry.
But
no,
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 it's fine.
good.
You and Jahan can
Oh, I
still
have
hang
to
on.
run
I have a one.
I got another call too.
So
But Tim, I just
not,
connected with you on LinkedIn, and
okay, perfect.
I hope we can continue some correspondence. I really appreciate your participation and thorough research that you brought to the Journal Club. It's just a great caliber, and I wish more people would come just like as prepared as you. It was great. It was really great. So thank you.
Well, since I do have, well, I don't know, I no longer have the hyperactivity as much as I used to, but if you send me the ADD or ADHD paper, I'd be glad to look at it.
Excellent. Yeah, I'll send you the yeah. I'll follow up. Let's follow up on LinkedIn. I have to run as does Teresa, but again, thank you, and
Yes,
great to connect.
I appreciate your time. I really do.
All right.
Thanks.
Well, bye for now.
All right, bye.